This video is amino acids backbone. The topic is macromolecules in the cell making and breaking. We're going to see that in one case water is removed and in the other case water is added in. And this impacts the ability for a bond to be formed or to be broken. As a reminder, there are 4 different categories of macromolecules in the cell, which you see up here. All these biological macromolecules are listed: proteins, nucleic acids, carbohydrates, and lipids. The ones that are most important for this course are proteins and nucleic acids. We're going to ignore carbohydrates for the most part, though you might encounter them in another biology course, like if you take energy flow. And then we're also going to talk about lipids, and in particular, we're going to talk about steroids and phospholipids in Biology 201. And notice that in this figure, you can have the function of these different sorts of molecules, and we can also see some of the representative small building blocks of their chemical structure. So all of the macromolecules that we just talked about are built through the same chemical process. They each have a small unit, which is called a monomer, which is repeated along the length of whatever the large macromolecule is. So macromolecule means large, and it typically refers to a polymer, which is something that has many subunits. And so each of those subunits is going to be bonded to the next, which we see here. One thing that's really important to notice here is that although each of these is just like a green rectangle, we don't see much details here. If we start to think about the actual chemical shape and the chemical structures, so they're not exactly symmetrical. So one side's going to be different than the next. And so we could think of this kind of like how we see these elephants that are in a line here. So each of the elephants would be a monomer. Notice that each of the elephants has its head oriented in the same location, and they're bonded to the next elephant in the exact same way. And so just like that, as we look at the chemical subunits, the monomers, and how they're bonded together to make this large, long polymer or macromolecule, they're going to have a very consistent sort of bond formation. So that's one of our goals, is to see exactly what that is here. And to talk through some of the text that I just wrote here. So basically, you can see that we have the polymer for many units as a chain of monomers. Monomer is one unit, and each monomer is going to be oriented in the same direction by chemical groups. Typically, the monomers are very similar, if not identical, to each other, and they're all bonded together in a row. Each of them is held to the next by a covalent bond. The process of adding a monomer to a chain is called polymerization, and so we're going to look at that in really specific detail. So here are the 2 different chemical reactions which occur that can allow a macromolecule to either be made or to be broken. The first one is called the condensation reaction. In another class, you might have heard of it as a dehydration synthesis. You can use either of those words, but your textbook likes to use condensation reaction. And so basically, just like the name implies, dehydration synthesis, water is going to be removed. So dehydration meaning like removing water. Condensation also implies that water is somehow collecting on a surface. And the synthesis is saying that through the process of removing water, we're going to synthesize something that is larger. And so basically exactly what happens here is we're going to check out that here's our existing polymer, and so we're going to add this new monomer to it. And so this incoming monomer, which we see here, it's got more chemical structure here that we're going to ignore, but on one side it has a hydroxyl group hanging off the side. It's got a hydrogen over here. So the hydroxyl group is actually going to be removed, so this bond is going to be cleaved, and at the same time, this bond right here, with this one little stray hydrogen that's at the very end of the growing polymer, basically those 2 are each going to be cleaved or cut at the same time, and so as they are cut out, then this forms water. So we've got 2 hydrogens and 1 oxygen. And so basically what that means is that we are removing water, and that means that each of these little green boxes now has the ability to form a new covalent bond, and so this is the bond that forms right here. That means that this incoming monomer is now part of the growing polypeptide chain, and here's the water molecule that's been removed. And again, this is the making of a macromolecule or making of a polymer is through the process of dehydration synthesis or a condensation reaction. So that's really useful for cells in certain times and certain places that they need to make large macromolecules. Other times they need to do some remodeling, and so then they might take apart those polymers. And so they're going to do that through a process called hydrolysis. Hydro means water, and lysis means to break apart. Like you might lyse a cyst or lyse an infection in your body or something like that. And so basically that word literally means breaking with water. And so basically we're going to break the polymer just a little tiny bit, doesn't all explode at once, but we're just going to throw one water molecule at this covalent bond right here. Notice that we've got our hydrogen and our hydroxyl group here. And so when it gets thrown at this little spot right here, then we're going to have a chemical reaction, so an enzyme is going to help do this rearrangement. This bond is going to get broken, and then the enzyme would add a hydrogen onto the end of the polymer chain. And then this sad little monomer is going to be looking for another bond to form. And so luckily, there's going to be a hydroxyl group that's hanging out right there. And so that can be covalently bonded to the monomer. And so basically here, again, we see that water is going to get added, the bond is going to break, and then we see a rearrangement of the water molecule as it's also broken. And so the monomer always gets the hydroxyl group, and the polymer gets the last, the hydrogen here to balance things out. And this will break apart the polymer. This, obviously, there still is a polymer here, but we could throw another water molecule at this bond, plop this off, throw another water molecule, plop that off. And so this is not exactly a very instantaneous process, but it's methodical. And just a little big picture, so this is the same process that occurs for making and breaking all macromolecules, whether they are proteins or nucleic acids or carbohydrates. All of those macromolecule categories use the same process.